These series of audio lectures are specially designed and created for educational purpose to reduce your eye strain and enhancing listening ability and stress-free preparation and revision. Chapter 2: Biological Classification. Introduction. First classification on scientific basis was given by Aristotle, who was the father of biology, who classified plants as herbs, shrubs, and trees, and animals as red-blooded anima and non-red-blooded anima, ANAIMA. Systems of classification. Linnaeus, 1758, gave two kingdoms of classification. Classification and the two kingdoms were plantae and animals. He placed organisms with cell walls in plantae and without cell walls in animals. Plantae was heterogeneous as it has unicellular and multicellular forms, autotrophic and heterotrophic forms, and prokaryotic as well as eukaryotes. Three kingdom classification proposed by Ernst Haeckel, 1866. As per the three kingdom concept, there were protista, plantae, and animals. Copeland in 1956 introduced four kingdom classification: Monera, protista, plantae, and animals. Five kingdom classification was given by R.H. Whitaker in 1969, which is the most popular. He classified organism based on cell structure, body organization, mode of nutrition, reproductions, phylogenetic relation. The main criteria was nutrition. The phylogenetic relation is also comes under cladistics analysis. Chlorella and Chlamydomonas, which were placed earlier in Plantae, and Amoeba and Paramecium, which were in Animalia, are brought together into kingdom Protista after modification. Six kingdoms of classifications. Six kingdoms classification proposed by Carl Woese in 1990 is based on 16S ribosomal gene sequence. Carl Woese splitted kingdom Monera into two new kingdoms. These are kingdom Archaebacteria and Eubacteria respectively. Kingdom Monerans. It has all prokaryotic organisms. Monerans are the most abundant microorganisms and occur everywhere. These are usually cocci, bacilli, spirillum, and vibrio or comma shape. The most abundant and prevalent form is bacilli, then cocci. It includes Archaebacteria and Eubacteria, Mycoplasma, Actinomycetes, etc. These are structurally very simple but complex in behavior and show most extensive metabolic diversity. Most of them are heterotrophs and show varieties of nutritions. Archaebacteria. These bacteria live in the most harsh and difficult environments such as high salinity, halophiles, acidic hot spring, thermoacidophiles, and marshy areas and rumen of the cattles, methanogens. These have special cell wall structure and plasma membrane structure, which is responsible for withstanding extremes of environmental conditions. Eubacteria. These have rigid cell wall and motile forms have flagellum. So they reproduce mainly by fission. The eubacteria comprises cyanobacteria and mycoplasma. Sometimes in unfavorable condition, they can form spore. They also show a sort of sexual reproduction called conjugation by adopting primitive type of DNA transfer from one bacterium to the another one. So that conjugation, transformation and transduction, these are possible. Pili structure helps in conjugation, and through conjugation breach, the plasmid or episome material can be transferred from F positive to the F negative strain. Chemoautotrophic bacteria oxidize varieties of inorganic substances and use the released energy from ATP production. For the ATP production, and they play a role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophs are the most abundant of all, and most of them are decomposers or saprophytes. These are useful in making curd, production of antibiotics, fixing nitrogen in legumes. Some are pathogen and cause varieties of diseases. Now, as we have discussed types of eubacteria, the first type is cyanobacteria or blue-green algae. They have chlorophyll A and photoautotrophs. These are unicellular, colonial or filamentous in structure, terrestrial or marine or freshwater forms. Filaments or colonies are surrounded by mucilaginous sheath and form blooms in polluted water. Some of this can fix atmospheric molecular nitrogen, example nostoc and anabinum, by having special cells called heterocyst. Mycoplasma. They lack cell wall and known as the joker of plant kingdom. They are facultative anaerobe and smallest living cell, also known as PPLO or pleuropneumonia-like organisms. These live as a pathogenic organisms in plants and animals. Since they lack cell wall, they are not sensitive to penicillin and cannot be killed by antibiotics and have pleomorphic form that can change its shape and called as a joker of plant kingdom as we have discussed. They have linear DNA. Kingdom Protista. It has unicellular eukaryotes and the boundaries of kingdom are not clear. A protistan may look like an animal or plant. These are primarily aquatic and form a link between the bacteria monerons and other kingdoms. They show both sexual and asexual mode of reproduction involving cell fusion and zygote formation. It has chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans as its types. Chrysophytes, these are the present in fresh water and marine environment and includes diatoms and golden algae or the desmids. These are passive planktons and most of them are photosynthetic and main producers of oceans. In diatoms, the cell wall is in the form of two thin overlapping silica wall is in the form of two thin overlapping silica shell like a soap box that's indestructible. Cell wall form diatomaceous earth over ocean floors. This soil is used in polishing, filtration oils, and syrups. Dinoflagellates. These are mostly marine, photosynthetic, and appear in different colors based on pigment present in them. Cell wall is with stiff cellulosic plates on the outer surface, so differ from chrysophytes, so it doesn't have silica. Most of them have two flagella, hence dinoflagellates. Di is two. Some dinoflagellates like Goniolux multiply rapidly and make the sea appear as red, known as red tides phenomenon. Euglenoids. These are usually present in stagnant freshwater bodies and have flexible proteinaceous pellicle in place of cell wall. They have two unequal flagella, example Euglena. They have chlorophyll A and B. In the absence of light, they predate on smaller organisms, hence shows mixotrophic mode of nutrition. MYXOTROPHIC. Slime molds. These are saprophytic protists and their body lacks cell wall. In suitable condition, they aggregate and form plasmodium, which grows for several feet. In unfavorable condition, the plasmodium differentiate and form fruiting body that have spores at their tips. The spores have cellulosic cell walls and extremely resistant and perennate. In unfavorable conditions, the spores are dispersed by air. Protozoans. All these are heterotrophs and live as parasites or predators and primitive relatives of animals. Protozoans are divided into four main groups: amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. Amoeboid protozoans live in fresh water, marine water, and moist soils. They put out pseudopodia to move and capture the prey as in amoeba. Marine forms may have silica shells. They can extend their pseudopodia from those shells like Alphidium, and entamoeba is parasitic form. But these shells are not made up of calcium carbonate. Flagellated protozoans are either free-living or parasitic. Trypanosome causes sleeping sickness, having flagella as a locomotory organelles. Ciliated protozoans have thousands of cilia, which are useful in movement and stirring the food or intaking the food into their gullet. These are dikaryotic. They have two different types of nucleus. Example, paramecium. Sporozoans have infective spore-like stage in their life cycle in organism like Plasmodium that causes malaria. So this is the end of the part 1 of the audio lecture. The lecture will be continued from Kingdom Fungi.